Okay, good day everyone. I'll just give everyone a couple of minutes to log in and join us, and then we will make a start. Okay, let's begin. First of all, thank you very much for taking time out of your day to join us today. I know we've got people from all over the world joining, so appreciate that there's different time zones and you're at different stages of your day. So thank you for spending a bit of time today with myself and Tyler. And I hope that you find the talk that Tyler's going to give shortly really informative and useful. This is the first of the ArrayJet 2021 webinar series. We're planning to run one of these at least every quarter, and at the end of the presentation today I will give you some details about the next topic and who our presenters are going to be. We're going to keep the format quite simple today. I'm going to run through a couple of slides round about ArrayJet and the partnership that we have with CDI, and then Tyler will come in and do his talk. During the slides, if there's any questions that you would like to ask, please use the Q&A function on the sidebar. If you press that, you can enter your question in, and then at the end we'll have a Q&A session where myself and Tyler will go through the questions, and Tyler will hopefully answer them for you. Okay, so with that, let's begin. So obviously it's Tyler from CDI Labs that's going to be speaking. I'm Gavin Boothroyd, ArrayJet's commercial manager for North America. Okay, and for those of you that are maybe joining us today that aren't familiar with ArrayJet, this is just a brief overview of what we do and who we work with. So we're an industry leader in non-contact biological printing. We offer a full range of solutions from instrumentation to contract microarray services. So we really can offer a full suite of options from instrumentation to services. The instruments that we offer are the fastest printing platform available in the market, but more importantly, we offer a modular instrument design. So this allows us to work with customers from academia at very basic R&D level, all the way up to high-end manufacturing customers such as CDI Labs, that are looking to print thousands of chips a year with us or with themselves. The other side of our business is the ArrayJet Advanced business. This is where we give customers access to our latest technology and also our team of scientists back at our world headquarters in Edinburgh. And these guys here would work with you to do custom array printing projects from R&D discovery assays all the way through to commercial biochip production. And I'll talk a bit about these two things more on the next slide and how that worked with CDI. But the main thing that ArrayJet really pride ourselves on, and I hope most of our customers would agree with this, we really work with our customers from initial contact through technical discussions and demonstrations, instrument delivery and after sales support. While we've got amazing technology, we feel our technology is only good as the people and the company that we are. And we really think that we are very good at delivering long-term value to our clients through ongoing support and work as they experience our instruments and services. So one of our best partnerships and longest established partnerships is with CDI Laboratories. So we've worked together for nearly 8 years now. The CDI Labs partnership began back in 2013. And for those of you not familiar with CDI Labs, these guys, this company produces the largest protein microarray in the world called HuProt. It contains around about 22,000 proteins on one chip, and this contains approximately 81% of the human proteome. So you can imagine that we require a service that, or an instrument that gives utmost precision and quality and spot morphology. So initially, when the CDI approached us back in 2013, we did some ArrayJet advanced service work with them, and this was used to do a tech transfer from their current printing solution, which was Contact, to ArrayJet's non-contact platform. And a lot of that work was carried out at our lab in Edinburgh. I know people from CDI came over and worked with us collaboratively, so to ensure that the product that we developed for our technology met and exceeded the requirements for CDI. Since then, CDI have grown rapidly, and they now have two of our high-end ArrayJet Ultramarathon 2s in their lab based in Baltimore. These instruments have the capability to print up to 1,000 slides per run, and this is very useful, obviously, for CDI's customers who are generally large pharma or biotech companies that are buying hundreds of these slides at a time and are looking for the consistency and the accuracy that the platform gives them by using our printer. Using ArrayJet's technology allows CDI rapid production of high quality microarray slides at time after time. And as I've said, the slides are made with high accuracy and reproducibility. It's also a really good technology because it means that their customers, when they're doing their downstream assay or their downstream work, have to use very little clinical sample, which can be important if it's a valuable samples that they're printing on. And the picture there shows one of the printed HuProt slides. So it's probably quite hard to imagine, but that's got around about 40,000 spots on it, the 22,000 proteins in duplicate. So you can see we're working on a very small scale. And again, that just reiterates why we need to have great accuracy and great throughput with our instrumentation. So now to hand over to Tyler. So Tyler's the Director of Immuno-Oncology and Biomarker Development with CDI. And today he's going to talk a bit about our technology and how it fits in with what they do at CDI, but also some of the other enabling technologies these guys have to carry out their work and the services that they offer. He'll probably talk about a few papers that they've been working on as well, and just give you a real insight and flavour as to what they do and what direction the company are moving in. So without further ado, can I hand over to Tyler, please?
Great. So I'm just trying to start up the talk. Oh, there we go. All right. So Gavin, does that look correct to you? Look good on your end?
That's great. Yeah.
All right. Sounds good. So I guess for those of you joining from Grace Bio, yes, this is the view of the Cascades from Broken Top. So a little bit about me. So I guess I kind of grew up and did my PhD out in Oregon. I'm still based here. I've been working for CDI for a couple of years, did an immuno-oncology PhD with a group out here, and started working with protein microarrays for like looking at IO samples back in grad school, which is kind of how I kind of ended up running into CDI. I wanted to kind of have an opportunity to continue that work. This is kind of just an image of our array overlaid over the stars. And so today I want to talk about a little bit about what we call the antibody fingerprint, or Seromics. Don't really know what the perfect term for this is as yet. I don't think the field's really decided, but it's kind of the universe of antibodies that your immune system bears. I mean, your immune system is a adaptive artificial intelligence that's able to observe, see, react to in a multitude of different ways, up to tens of thousands or even more targets simultaneously. How this all works together, what this means for our health, I don't really think is determined as yet. And so I think what's so exciting about working at a group like CDI, which was primarily started as a proteomics company and has since kind of migrated to more of a immunology type profiling business, is, you know, there's a handful of groups in the world like ArrayJet, like Grace Bio, that are making either the films or the printers that allow us to actually go after these sorts of things. So some of the data that I'd like to show most to you right now, I'm actually unable to due to some collaborator contracts. But essentially, like the gist is all of us have a very unique fingerprint of autoantibodies. They're very stable over time. And I think this has some impact on our health. And I'll show some examples of that, both with our technologies and some other ones. So I'm actually going to kick it off with our non-array platform, because I have a little bit more rights to show some of this data. So I'm going to try to see if I can move this over to make sure it's not blocking our view of this. But this is another high throughput platform that we have called VirScan. This is out of Steve Elledge's group at Harvard. And what this allows us to do is look at the universe of antiviral antibodies. We also have a human library for this as well. So essentially kind of the way that this technology was created was a bunch of sequences were downloaded from HuProt covering the majority of the known universe of human viruses. These were created into proteins, which were chopped up to 56 more peptide tiles and packaged into T7 bacteriophages. This has gotten kind of a lot of popular press, kind of similar to some of our more recent HuProt work. And so the way that the assay is run is there's about 100,000 viral peptides in the viral library. We've got about 250,000 human peptides in the human library. You react those phages with the patient's serum. You do a protein AG pull down assay. You grow up the sample, you pool them together, and you run them on a sequencer. And you're able to get like a counts result for how many times was X phage identified in the sequencer result versus Y phage. You compare that to some protein AG beads only controls. And so what you're able to get is like a readout. So this assay in general, we think is a little bit less sensitive than our HuProt microarrays, but it lets us look at kind of a broader swath of content. And I'm able to show some of the data just to give you a feel for what are we talking about? How many antibody responses are we talking about with the antibody fingerprint? Because depending on what citations you look into, it's actually quite broad. So this is a set of about 200 samples that were run on our both VirScan and HuScan and our ZScores xenoantigen libraries. These are all for pediatric patients, either controls, ones that are actively just altogether COVID, either under acute or convalescent. MIS syndrome, this is a hemorrhagic autoimmune disease, immune thrombocytopenia, juvenile arthritis, lupus, autoinflammation, ulcerative colitis, Crohn's disease, just looking at the total number of hits in both the viral and the human library as defined as, by significance versus the beads controls. So you're seeing that in all these samples, everyone's got hundreds of unique memory responses that are really strong enough to be identified by this PhIP-Seq phage display library. So kind of break it down to just kind of human versus viral, just to kind of have an unbiased look of, what does this universe look like? So there are fewer peptides in the viral library. You see that almost everybody universally is having more hits to the viral peptides. So the control kids are having, maybe 400 responses on average. Maybe it's around 50, 60 to the human library. But this is really interesting to think about because these are really strong, really high affinity autoantibodies. And even in totally healthy children that are not having autoimmune disease, there's lots of background autoreactivity. And so what does this mean for health? I think, or exactly what this means for health is not entirely known, but I'm going to show some examples where I think it's pretty clear that it's very impactful and something that's worth going after. And so us at CDI, and in particular what we call our antigen services, I think we have the most powerful tools yet existent in the world, both with our microarray, which is kind of the old school technology that the company was founded on, as well as the PhIP-Seq libraries, which is a bit more content, a bit less sensitive, doesn't have the isotype specificity the arrays have. We have two different, completely orthogonal approaches at looking at this and figuring out what it means for immune behavior. So since these are COVID samples, and this is the MIS syndrome, this was formerly called Kawasaki-like, you see that this is probably just due to there being more global antibody. So I imagine that our pipeline is not totally normalized for just cutting out the difference between higher versus lower, at least in this case. But to give you kind of an idea of what this means, say we zoom in on one protein. So now we're taking the 300, or excuse me, it's not 350,000 proteins. It's about 50,000 proteins in the assay, 350,000 peptides. We now zoom in on one of those proteins and several dozen peptides within that protein. This is just a random human coronavirus spike. And what we see is with a number of the kids that have either acute or convalescent COVID or have the MIS syndrome, they all have a couple of epitopes that they're making really strong antibodies to that the healthy kids do not. We see that with another unrelated spike protein for the coronavirus. We see that also for old school SARS. So they all had COVID-19, they all make autoantibodies to the unrelated spikes. And I guess just as a topic of interest, because we're all going through the pandemic right now, everyone's worried about existing strains and whether or not they're mutating to spread faster. What we see here are viruses that are millions of years and cross species separate from COVID-19, and people are making really strong antibody responses to the spike. And that's because, well, the human ACE2 receptor, the mammalian ACE2 receptor is pretty stable. The virus has to bind that. And therefore, potentially there are epitopes that cannot be evolved around. And so kind of my great hope is that what's coming up with the vaccine, and there's some additional studies from Pfizer that potentially their vaccine is cost reactive to emergent strains. I don't think that COVID-19 can evolve around this, and it's visible right here in kind of our own little independent data set. And so to me, I find that really interesting and really powerful that, you can zoom in on this immune behavior and potentially see things that you might not be able to see otherwise if you weren't scanning so much data. So to kind of start zooming out at this concept in general, like what do we mean by the antibody fingerprint? So I already said before that it's really stable over time. So there's kind of this old paper by Bob Nagel that was done, who's now a collaborator of ours with HuProt, but this was done with protearrays back in the day. So Invitrogen used to make a product with about 10,000 proteins with pin printers. And these are the ones that I actually used back in grad school. That was a lot less coverage and content, but a lot of existing work was with that. Now, these arrays seem to be a lot more reactive. Bob actually found in this paper that as much as 10% of the human proteome had autoreactive antibodies. We do not see that with HuProt. We see maybe like 10, 15% of that, maybe a couple hundred per sample, similar to what I was showing you here in our HuScan data. Once again, these are kids, and so they don't have very many. If this was an adult or an aged population, this cluster would be higher up here. So this is kind of a core feature that was published in this paper, and I'm hoping we'll have other people that have kind of a larger overview of this that we can give them the time in the future. But essentially, as you get older, you have more autoantibodies. The older you are, the more autoreactive the antibodies you bear. This is probably due to a lot of things, such as just natural genetic drift as a human being, right? So some of your cells are going to start looking less like other cells, which is how cancer arises. It's also just you're going to have more opportunities for accidental cross-reactive experiences. In general, women have more total auto IgG than men, but men have more IgG4 and IgG4 related diseases. It's generally thought that the IgG4 FC receptor is somewhat inert, might be a somewhat tolerogenic, non-cytotoxic autoantibody. And so there's maybe some of the sex differences between things like, say, COVID-19. If there were to be an autoantibody related link, you would see that autoantibodies are associated with age and sex. And I'll show a paper that potentially suggests that with one of our collaborators a little bit further on. So to kind of give an idea of what I think is the opportunity here from an evangelism side. So this is the top NIH budget items in millions of dollars. So this is 15 billion on clinical research, 10 billion a year being spent on just pure genomics, and then a bunch of sub-disease breakdowns, things like cardiovascular disease, heart disease, cancer, diabetes, stroke, Alzheimer's disease, alcoholism, a lot of things that we know kind of some of the background risk factors, but we don't really have a good control over the underlying cause. A lot of these things are stochastic, age-related, lifestyle-related, but what does that mean in the biology, right? Like your body, it doesn't just know you're being, just because you drink too much or you eat too many carbohydrates, it's not perceiving that you're immoral through the brain or something like that, right? There's actual biology that's going on. So much genomics has been done in recent years. It's pretty clear that almost all of these conditions are not Mendelian genetic diseases. I mean, there are some cases of familial Alzheimer's and such, but in general, it's not one disease, one condition. There's a lot of different factors. These generally get lumped as multifactorial cases, but there's always people that do all the wrong things and don't get the condition, right? So how do you identify that? Is there something in that that can help us lead to better treatments or biomarkers that lead us to the underlying conditions? So more and more over the years, a lot of these conditions are increasingly being recognized as quote unquote inflammatory diseases. What's interesting is immune behavior is very stochastic. It's age related, it's lifestyle related, and it's measurable. And we see a lot of things like, say, the amount of autoantibodies in human beings that seem to increase with age, and a lot of these things that also increases with risk for cardiovascular disease and regenerative disease. Is it really just the cells getting old and getting broken, or is it the immune system has more opportunities to become skewed over a lifetime and then potentially starts causing chronic problems? I'm of the personal opinion that the immune system is probably doing more than we currently recognize it. And it currently does. And that's essentially what we're trying to accomplish with some of the tools that we provide to help people kind of unravel these things as we go forward. So when we talk about inflammation, probably the example that I have the most to understand about, or I have the most experience with personally, is with cancer. So when people call it inflammatory disease, a lot of times they're taking tissue or they're just looking for immune cells and histology. So to give kind of a concrete example that's a little more simple with that, so these are some mouse experiments I did before joining CDI. So this is a simple cancer vaccine experiment with what's called a 4T1 mouse model. So that's a mouse mammary cancer, so a simulated breast cancer in the mice. You give the vaccine. This is an autophagosome vaccine that's currently undergoing clinical trials at the institute I used to be at in Portland. You boost, then you either take cells out to characterize or you implant tumors and you see what happens. So if you add vaccine plus poly-IC is the adjuvant. So add the vaccine plus the adjuvant, you get improvement in survival, the tumors grow slower. These separations aren't really huge because this is a highly virulent, really effective tumor. To really see a separation at all with 4T1 is pretty impressive. And so what is going on in this case? Well, so here's some multispectral histology images of a tumor that's then been growing for a while, either with just the adjuvant only, or they got the antigenic experience. And all these little red dots that you're seeing are CD3 positive, CD8 positive killer T cells. So you induce an adjuvant, you alter the downstream inflammatory event. There are immune cells in there that are causing changes to the overall biology. So this is very clear in the IO space. It's very clear in cancer. And it's kind of my personal opinion that stuff like this is going on in a lot more conditions than we think. People are reporting on this, but they generally don't go to the quote unquote antigen level. They're just kind of seeing that immune cells are there and, oh, isn't this neat and interesting? And I think to really start solving issues, you have to figure out what are these immune cells doing? And so in this case, we knew what they were doing because we knew what the mutations were in the mouse model, and we did a variety of things that we're not going to talk about today. But essentially, you have an immune microenvironment where there's cytotoxic T cell killing, you have antibody-dependent cell killing with macrophages and NK cells versus, say, a more tolerogenic environment, perhaps with some IgG4 antibodies that are helping protect the tumor, T regulatory cells. And so what you get is a difference in the overall inflammatory environment. And so when people are talking about a lot of diseases being quote unquote inflammatory diseases, what they really mean is it's an immune behavior. And immune behavior, as we know, is generally an antigen-driven phenomenon. So it can be like a local and direct antigen. So the tumor can directly be dying, releasing protein. It can be cross-present. It can be activating the immune system, or it can be a distal cross-reactive site. So a lot of press is given to the microbiome these days. That's because like 60, 70% of all your immune cells live in your gut. And a lot of what happens there then translates to what is going on in other areas of the body. So it can be like a distal cross-reactive effect, or it could be something that's entirely historical, right? So you could have had a viral infection in the past that potentially triggered your immune system to be biased for some sort of more incorrect response going forward. And so what's really interesting is those autoimmune kids that I showed you earlier, we're seeing a really high prevalence of EBV or Epstein-Barr virus or mono in a lot of those kids, which is actually a little early for a lot of those kids to have mono. It's usually thought of as a kissing disease and not something that you'd be getting before puberty. And so it's interesting to see that all these pediatric cases potentially having had a historical viral event that then triggers a downstream event where potentially they're predisposed for these different autoimmune conditions. And so kind of there's the whole universe of ways that either that this immune experience can occur. It can occur from foods and the microbes that grow on them. It can occur from natural infections. It can occur from vaccines. Like, yes, vaccines are generally safe, but every time you inject adjuvant into the human body, there's going to be human proteins around there. There are opportunities for things to occasionally go wrong. Your differences in your HLA receptors, whether you're male or female, predisposes and biases your immune system to different behaviors. And then, of course, your lifestyle. So when you exercise, it releases DAMPs. It's generally thought to be tolerogenic. So damage-associated molecular patterns, so damage signals without adjuvant being released from cells seems to calm down the immune system, teach it to behave. All of this across your lifetime kind of accumulates as what we call the immune fingerprint. And so that fingerprint is created with both T cells and B cell or antibody producing plasma cells or B cells, it's a lot harder to measure the T cells in mass. And so these are two from our colleague, Steve Elledge, two different technologies that are going after that. He does have a way to profile T cells in mass that he's reported on. Now, the challenge of this is it involves flow cytometry, and they have to create a different HLA clone because this T cell receptor has to be bound to HLA. So there's actually the peptide inside of the MHC1, MHC2 that the T cell then recognizes. And you'd have to make thousands of different versions of this assay to actually profile the diversity of human T cell activity. And it's just not feasible at this time. And nobody is offering something like this. Either computational methods or other things in the future will hopefully do this. So in the meantime, if you just have the protein libraries, you're able to directly measure antibodies. And so that's what we do at CDI as a proteomics company, is we have all these massive proteomics libraries that lets us go straight to the antibodies that are produced by the plasma cells and see what they're doing right up front, both with this technology from Professor Elledge and with our own that were created at Johns Hopkins. So to give a couple of really potent, some of my favorite examples of how do we know that it's not just the T cells that matter, that the antibodies are in there doing very specific, interesting things. This one's something that T cells certainly can't do. It's actually affecting immune signaling. So I'm going to zoom in on these two papers separately. So this paper is air deficient patients harbor unique high affinity disease ameliorating autoantibodies. So for those of you that aren't familiar with AIR, this is a systemic autoimmune disease. AIR is a protein in your thymus that helps teach your T cells tolerance. And so if you're an AIR knockout, you're doomed for all sorts of autoimmune conditions. In this case, the kids seem to either get or not get type 1 diabetes, so autoimmunity against their pancreatic islets. Well, in this paper, it turns out, and these were identified from protoarrays, so a protein microarray technology very similar to our HuProt chip, that the kids all had autoantibodies to interferon alpha pathway cytokines if they didn't get type 1 diabetes. So there's some sort of immune event that they had in the past. It induced anti-cytokine autoantibodies that have interfered with immune behavior. And what that resulted in was the pancreatic islets not getting destroyed. And so there's something in that antibody fingerprint that completely altered the biology and quality of life trajectory for these children. Now, what I think is really exciting and really interesting is something was just reported related to COVID-19 infections. So this is out of a really large consortium, including Steve Holland out of the NIH and his postdoc Lindsay. And what's really fascinating is they go through patients that have life-threatening COVID-19 versus asymptomatic mild, and look at the N's here. N of 987, 663 on this side, looking at how frequently do they observe anti-interferon, type 1 interferon autoantibodies. And what they found is that all the patients that did really bad have these pre-existing antibodies to interferons. And so now we don't know whether that's correlation or causation straight up, but it's kind of my hint that it's kind of my thought that potentially it is. And this is a really fascinating thing because what do we know about autoantibodies? Well, they increase with age. Children have very few of them whatsoever. And all these other factors that kind of overlap with what we know about COVID. There's also a sex difference. And so I guess they didn't go and look at the specific isotype, but based on men having a lot more IgG4, maybe this is a more IgG4 type autoantibody, but they didn't look into that. They just looked at total IgG, I believe. And so once again, whether or not the lungs are protected or destroyed is somewhat at fate of what the autoantibodies were doing prior to the disease occurring. And so when you kind of come back to the NIH list, you have a lot of different conditions, and we kind of start to wonder, and a lot of our clients that we work with are starting to wonder whether or not maybe some of these are antigen-specific immune conditions where we can either find a biomarker or a directly interfering something that is a signaling molecule that might be affected by an autoantibody that might might help us decide whether you fall into group A or B for high risk or low risk, and whether or not that might lead to potential opportunities for new diagnostics or drugs. And so I personally have the opinion, this might not be right, that when we start thinking about a lot of these quote unquote inflammatory conditions, whether or not you get plaques in the arteries, whether or not your brain has problems with Alzheimer's disease, whether or not a developing fetus develops autism. There's no genetic link really to autism, right? What you see are the older the mother is, the kind of like there's several other risk factors that seem to associate with the sort of thing that would increase the odds of having autoantibodies. You wonder whether there's some sort of inflammatory response against the fetus, and there's been some papers that have shown stuff like that in mouse models. And so kind of it's our thesis here at CDI Antigen that inflammatory often means some sort of undiscovered antigen-specific immune disease, which may or may not involve antibodies, but there's obviously going to be some sort of signal, we think, in some of them. And we think that a lot of these technologies that we have are allowing people to screen for these things and maybe kind of find the answers to some of the conditions for the first time. So it's been really exciting. So even take like aging, for example. So some people, women live longer than men. Women also get more autoimmune disease than men. Some women live a lot longer than others. Like we have people that seem to live really healthy to 110. We have ultracentenarians. Part of me wonders whether, could you go into the antibody fingerprint of these people and see maybe a tonic autoimmunity? So there's a really interesting group. So Michelle Settlein out of Memorial Sloan Kettering. So one of the kind of original CAR T cell or chimeric antigen receptor T cell receptor groups has done a set of mouse experiments where they've actually done a CAR T cell against senescent cells. So they found a biomarker in mice that identifies senescence. They are raising CAR T cells against that. And it's essentially an anti-aging immunotherapy where the T cells go in, kill the senescent cells, the mice then live longer. I wonder whether our sorts of platforms could help identify additional targets for going after those sorts of conditions. So I think it's a really exciting field, and it really isn't explored all that much yet, in part because the tools haven't existed until very recently. And so those tools are built by us here at CDI. So we've got about 30 employees or so. And so we were founded by a number of Johns Hopkins faculty, along with our CEO, Ignacio Pino. It's been expanding over, almost a decade or so. We've got a facility in Baltimore that's still, we're just now moving out of our startup incubator at Johns Hopkins, moving into a newer, larger facility across town. And we have a large lab team down in Mayaguez that produces proteins, and the arrays are all made up in Baltimore. So we have a lot of really interesting people that are involved with this. So Jeff Buca, who actually currently leads an international synthetic yeast consortium where they're trying to actually make an entire yeast from scratch, is now director of systems genetics at NYU, along with Hung. So I think I was about to show, yeah. So Hung actually co-created the original proto-array. So you've heard me say about proto-array a couple of times. This is an older protein microarray technology. So when Hung was with Michael Snyder at Yale back in the day, so Mike Snyder is now at Stanford, but they created the original HuProt chip. He then became faculty at Johns Hopkins, and Xiaowei Hu, who is now our proteomics director, worked with Hung to develop the HuProt chip. And so the HuProt chip, as you see some more beautiful images of it being printed on our ArrayJet printers, was originally created as part of a large NIH program project grant called the NIH Protein Capture Reagents Program. So the array was created as part of like a $15 to $20 million grant to develop this array to detect cross-reactivity to make really good monoclonal antibodies against transcription factors. And so you can either go on our website or you can just kind of look up this link and see kind of the way the array was validated. So the way that the array was produced, the technology that allows us to look at these antibody fingerprinting results, is we got these large ORF libraries. Hung and Xiaowei and other team members in those labs cloned them into these vectors, which were then transformed into yeast. Colonies were picked. They were sequence confirmed as part of this Nature Methods paper. And then about every year, so maybe every 15 or 20,000 arrays or so, we grow up all these clones to create protein. So each of the proteins has a GST or a glutathione S transferase tag on it that we use for purification to separate our proteins from the rest of the proteins in the yeast. We confirm by gels that some percentage of these are successful. We then print them on our chips. And for each batch of arrays, we use that GST tag to confirm that the arrays actually printed. And so we kind of have this whole round trip process where you start with the ORF, we've sequenced each clone, we know GST ends up on the spot, and that's how we kind of round trip know that when we're giving you a protein microarray result, that the target that we've claimed to have identified is actually the one that we think we identified. And so when we do an actual individual assay, it's really quite simple. You take some sort of sample that can bind to the material on the array. We do also do DNA binding and protein binding, but kind of the core focus and the largest part of our business is on serum. So all isotypes of antibody are available in the serum. They will bind to the proteins on the array, and then we just come back with whatever secondary you're interested in looking at. So IgG, total IgG, IgM, and IgA are the most common ones, but we've kind of got some experimental activities going on where we're trying to look at doing more multi-isotypes. I think I was talking about IgG4 a little bit earlier versus maybe the more cytotoxic IgGs. And so wondering whether or not some of the differences there might lead to differences in outcome, because almost all papers on protein microarrays and antibody fingerprint right now are total IgGs. And that's kind of unfortunately, one of the current limitations of the PhIP-Seq, VirScan, and HuScan assays. So there's a protein AG pull down, so that gives you all of the isotypes all at once. And the isotypes have different FC receptors. They have different biologic activity. They do very different things. And very little work has yet been done to kind of untease this at a proteome-wide scale. And it's something that we're really excited to be working with people on over the next few years. Kind of to give an example of kind of what a fingerprint of a single healthy human male looks like. These are a number of different arrays printed across different batches with these ArrayJet printers. These are the ones that are from the individual batches. You can see that the correlation coefficient on these log plots is really tight, like 0.98, 0.99 for both the green and the red channels. So the red is IgG, I believe the green is IgA, essentially showing both the reproducibility of running samples and the reproducibility of the print quality that we're getting from ArrayJet. So kind of to talk about our clone library. So we have the majority of the human proteome. So because these were created essentially from public WRF collections that our founders obtained, it was a lot of stuff that was of most interest and being researched at the time. And so even though we're missing some content, it's a really, really good overview of what's available in the human proteome. It's very rare that I'm looking for a target and I end up not identifying it on the HuProt array, regardless of what the category is. Another nice thing is that, well, yes, membrane proteins, we can't guarantee that they're perfectly folded. Because there's that GST tag on there, it helps. We have a soluble protein attached to each protein, and that seems to help us solubilize and recover like receptors and such. And so we do think that we definitely do see like anti-receptor autoantibodies identified on HuProt at times, which I think is pretty cool. We also know that a number of them are well folded. So this is a poster done by Xiaowei and Hung a number of years ago that essentially shows that if the kinases printed on the array will autophosphorylate. So we at least know that, you know, with some protein classes that there is activity. There's lots and lots of groups that have been publishing on the use of this technology, probably majority autoantibody profiling over the years. And this has definitely grown since I made this slide about 18 months ago. Kind of one of the more interesting partner projects that we have, I guess, directly related to the IO space, is with a collaborator of ours, Iman Osman, where we've been using these microarrays with a large cohort of ipilimumab, nivolumab, so anti-PD-1, anti-CTLA-4 responders, and it shows that you can see in the baseline autoantibody profile, so that autoantibody fingerprint, there is a kind of a set of autoantibodies to proteins prior to therapy that seems to predict toxicity to the therapy. And you know, we know the arrays, as I was just talking about, anti-receptor autoantibodies, after the patients get anti-CTLA-4, the CTLA-4 spots light up, which is kind of interesting. So this is the original paper a couple of years ago. We have a much larger cohort that was just presented at ASCO last summer, and that's even being expanded a little bit more. And so we expect, I don't know if that'll be, hopefully be out later this year, hopefully a really nice publication kind of demonstrating this. And I've also played around with the data a little bit myself. So this is a poster that I presented at SITC, or the Society for Immunotherapy of Cancer, fall of 2019, essentially just kind of looking at the overall, like stop looking at the individual targets, but instead of toxicity, looking at responses. And you know, that it's not as many samples and the differentiation isn't quite as good. But what's interesting is we do see a couple of global differences in the overall responses. And so essentially what I'm talking about with Z-scores is looking across the cohort. If you're assuming that most patients don't respond, does this patient have a response to that antibody that is unique above everybody else's? And then count the number of unique responses that that person has in their antibody fingerprint. So kind of using two different thresholds, either looking for really, really bright ones or kind of dimmer ones. And what we see is with the dimmer ones, with the patients that do worse, they have a lot more background autoantibody binding than the patients that do well after therapy. And now it's not significant on the PD-1 side, but we see kind of the opposite. It's kind of interesting thinking about CTLA-4 maybe being a releasing, boosting therapy. There's maybe a little bit more, oh, excuse me, that's PD-1, saying it backwards. CTLA-4 potentially inducing new responses. There's maybe a bit more immune space here, whereas this maybe is a bit more disorganized. There's a lot of background autoantibodies that maybe aren't binding their tumor that destines these patients for kind of differences in outcome. But it's going to be interesting to dive through the larger cohorts when they're here. So that's been 30 minutes, which is when Gavin asked me to kind of wrap it up. I do have more slides, but I wanted to hit the 30-minute mark so that we could go through and have some questions. So,
thank you very much, Tyler. That was really, really good. Just put the screen back up. Okay.
So I'm seeing the first question about long COVID.
Sure.
Almost certainly, we have several groups that are in contact with us right now that are talking about initiating long COVID studies, and whether or not there's some sort of trigger that has been created by COVID. I guess one of the challenges is, to some degree, is with that type of study is ideally you'd want to see what their autoantibody fingerprints look like pre-COVID. Like you'd want to see what sorts of changes were induced. And I imagine that most of these studies, I think most of the studies that are coming in with us right now are just looking at patients that ended up with long COVID versus didn't after COVID infection. And while I think that'd be interesting, you'd probably take a lot more samples to figure it out. So if you have access to pre-post serum, like before COVID infection, and then some of those ended up either getting or not getting long COVID, I think that'd be a really interesting cohort to potentially try to begin to tease that out.
Yeah, one question I've had come in, Tyler, is, so obviously you've got a number of different assays and platforms within CDI. Do any of them complement each other? Do you ever validate results in one technology and look at it in a different one?
Yeah, so kind of give a little bit more background on the PhIP-Seq. So we just got the license on that signed like in January of 2020. We started running our first assays in June. And I think we've run maybe 1,500, 2,000 samples. We have a couple of really large studies in the pipe that we're about to do. So we don't really have as good of a feel for the technologies we'd like to. But I do have several where I've got both platforms, and they give somewhat overlapping, but a bit more different results than you'd expect. And the reason that we think is the HuProt assay is it's a little more analog, where you get fluorescence intensity of from 0 to 65,000 on the scanner. And so you could see these really subtle changes in autoantibody binding between patients, where the PhIP-Seq, it's a capture and pull down assay, and then you literally have to get readout counts. The dynamic range, like so a screaming PhIP-Seq hit might be 300 copies of phage recognized versus say like 20 or 30 for the controls. And so I think you're just seeing the much stronger responses there. And so I think that they're kind of detecting these kind of orthogonal, they're kind of very different types of platforms. They're seeing different things. Now, that said, kind of my hope is, and some of the stuff that I'm kind of working on is, we do have, because the PhIP-Seq is peptide tiles, right? You can maybe start to look at cross-spreading of the response and multi-epitope responses to proteins. And obviously, if you see hits in both totally independent platforms, those are ones that I would trust the most. And so I'm especially if you're trying to incorporate maybe some like overlapping T cell information, like I'm trying to do some stuff with overlapping like net MHC2 predictions with the PhIP-Seq data, because now you can start to identify which are the stronger epitopes. Does some of those epitopes overlap with what we know about T cell behavior? And can we also see that on HuProt? And so, I mean, I guess that it's a bit of a roundabout question to say that we don't totally know how well it meshes together, but in theory, it does really well. And I've been really enjoying looking at the overlap. So.
Okay, we've had another question come in. So it reads as, COVID has also shown differences in severity of symptom fatality linked to ethnicity. Have you looked at any ethnic differences in antibody profile and how this might contribute?
Yeah, so there were definitely, so in our healthy women cohort that unfortunately I wasn't able to present the data today, we do have like an African-American subcomponent and some other ethnicities. The issue is these are all healthy women, and it's kind of a scattering of ages. And so there wasn't obviously any ethnic differences, but it would be my kind of personal hunch that things like the number and background of autoantibodies would probably correlate with some of those ethnic differences and potentially would contribute to that. So things like, just even skin tone and vitamin D absorption, or whether it be cultural dietary differences, I would not be surprised at all, but I haven't seen examples of that data personally as yet.
Okay. Has anyone else got any questions? If so, please hit the Q&A button or the chat button. We'll just give you a couple of minutes. Nope. Okay, I think that might be us. So Tyler, thank you very much for your time today. That was a really great presentation you gave us. Hopefully, maybe once you have more data to share later in the year, we could maybe get you back for a second webinar to talk a bit more detail about that.
Yeah.
I know you're excited about some of the things that are coming up. I'd just like to thank everyone for joining today. Our next ArrayJet 2021 webinar will be on February 10th at 1 o'clock UK time. So that will be 12 o'clock Central European time. Trying to do some maths in my head. 8 o'clock EST in the US, and 5 AM in Pacific. So that's quite early. But like all of our webinars, they will be recorded and available to watch on demand after the broadcast. So the next webinar that we'll be doing will be glycoprofiling using carbohydrate microarrays in novel applications. And again, it will be a couple of ArrayJet customers that will be speaking. It will be Professor William Willits from the University of Newcastle and Dr. Sylvia Vidal-Melgosa from Max Planck in Bremen. Please do feel free to register for the webinar now. I believe that it's been put onto the front page of our ArrayJet website today, and it will be similar format to this where you register and you will receive the details through closer to the time. Just once again, just on behalf of myself and Tyler and the teams at ArrayJet and CDI, we would just like to thank you for your time today. And yep, thanks very much, and we hope to speak to you soon. Thank you.